Okay, welcome everyone. I'm Jennifer Smith, a science communicator with Wisconsin Sea Grant based in Madison. This is our third lake talk of the fall 2020 season. We started offering these talks in person earlier this year before the pandemic began. Now, for obvious reasons, we've moved to a virtual format. So thanks so much for being here with us tonight online. We're glad to have you here, and we're also glad to have our two speakers for this evening. It's Katie Olson and Brandon Fallish, and I'll tell you more about them in just a minute, but first I'll go over a few housekeeping details. This session will be 1 hour, and we'll have time within that for your questions. And when we get to the questions, the best way to submit them is to use the chat function. I know many of you are familiar with Zoom already, but if you mouse around near the bottom of your screen, there's a little chat box. So if you put your questions in there, when Katie and Brandon are done with the presentation, I'll share those questions and they'll be happy to answer them. If we have more questions than we have time for, we can follow up on the Sea Grant blog or some other way to get those questions answered. Also, this session is being recorded in case you'd like to watch it later or share it with somebody else. When that recording is ready, we'll put it on the YouTube channel for Wisconsin Sea Grant. So the best way to stay in the loop about that recording or other events like this is to follow Wisconsin Sea Grant on social media. So you can find us on Facebook and Twitter at the handle @UWISC-SeaGrant. Now I'd like to give you some information about our two presenters, Katie Olson and Brandon Fallish. They're both graduate students at the University of Wisconsin, Green Bay. Katie works on the benthic macroinvertebrate communities of Lower Green Bay and the Lower Fox River. She's a graduate of UW-Stevens Point, and she has a degree in fisheries, and she also minored in biology and water resources. After college, she worked as an aquatic invertebrate biologist in northern Minnesota before she came to grad school at UW Green Bay. Brandon studies environmental policy and planning, and his thesis work is connected to the health of the food web in Lower Green Bay. Before he came to graduate school, he worked as a fish biologist for both the U.S. Fish and Wildlife Service and the U.S. Geological Survey. So thanks again to Katie and Brandon, and now I'm going to turn off my camera and turn it over to them.
Hello, everyone, and thanks for the introduction, Jennifer. Before we get going here, I would really like to thank our funding sources as well as the other people listed on this slide. So a big thanks to Wisconsin Sea Grant, the Wisconsin DNR, and the Environmental Protection Agency for supplying the funds for our work. It would not be possible without them. And everyone else listed on this slide has made this experience truly enjoyable. Our research has been so much fun, and thank you so much for all of the help you've provided us. So for those of you not from the area, this is Green Bay. I'm going to be referring to the following regions a couple times throughout this presentation, so I thought I would take some time and point them out. The Inner Bay is the southernmost part of Green Bay. It's outlined here in red. It's also known as the area of concern. The Lower Bay is the region just north of that, and it extends to the entrance lighthouse, which is the yellow dot here you see in the center. And everything north of that up into Sturgeon Bay is called the Middle Bay, and that's outside the scope of this map. The bay is 99 miles long before it dumps into the northwestern part of Lake Michigan, and it's about 14 miles wide. It also drains 10.6 million acres of land, which is a crazy amount. And the water quality is largely driven by the Fox River, which is the southernmost tributary that's partially outlined here in red, and Lake Michigan. And so with that, Brandon's going to discuss some of the historical uses of this unique watershed.
All right, so we're just going to go over the history a little bit. So just kind of back us up to about 10,000 years ago. The bay and Lake Michigan and this whole area sat about 25 feet higher in elevation than it does today. Today it sits at about 581 feet above sea level. Then it was around 605. So the bay didn't exist back then. There wasn't a bay. There was a larger lake that they called Lake Chicago, which is now Lake Michigan. And the bay formed around 4,500 years ago. So fast forward to about the 1700s, French settler Charles de Langlade was the first person here in 1745. He was booted out by the British in 1763, and then they got the boot by the Americans when we won the Revolutionary War in 1776. So at that time in the 1700s, the bay was rich with life, rich with wild rice, grew rampant. Wild rice was the dominant plant type in the bay, and now it barely exists. We are trying to do some efforts to bring it back, which Katie will touch on later, but we can't get it to grow naturally right now. So by the 1700s, Green Bay was the largest fur trading port in the eastern United States. So it's a very large city due to the port. And at the time, there was no detriment to the water habitat at that time. The only real detriment to the ecosystem was the fur-bearing mammals. As I said, there was the largest fur trading post in the eastern United States. So a lot of animals were being killed in that time. So fast forward up to the 1800s. So this is where we start to go up you know what creek without a paddle. Everything goes downhill from here. This is what I like to call the age of destruction. In 1810, the lumber industry comes in. In 1841, land sales commence from the United States. In the 1850s, agriculture joins in. In the 1870s, commercial fishing really starts to take off. And if that's not enough, in the 1890s, the paper and manufacturing industry decide to join the party. So I'm going to dive into a little bit deeper to each one of these things. So the lumber industry, lumber industry started in about 1810. They couldn't legally lumber at that time, but they were just doing it anyway because there was nobody to stop them. There was no regulations. The government did eventually give them permits, but they weren't within their legal bounds at that point in time because they didn't technically own the land yet, because this was still in the transitional period in which we were, acquiring the land from the Indians. So in 1850, the United States produced about 5 million board feet per year, and that jumped up to 40 million board feet per year in 1907. And if you see in the graph on the right here, I just kind of, Wisconsin and Michigan in 1880 accounted for over 30% of the lumber production in the United States. So that's a lot. And they like to be near water because it was a lot easier to transport wood if they were near water. So they concentrated their logging operations next to the Fox River. So there was this mass clear cutting on both sides of not only the Fox River, but every river in the state of Wisconsin. And that led to this increased sediment and nutrient load in the river system, because now before you had these trees, that is this intertwined matrix of roots that's holding nutrient load, taking up nutrients, holding the sediment where it is, and now you just clear cut all that. That's like open the floodgates to the river. So now you just have this nonstop flow of nutrients and sediment load created by the lumber industry flowing into the river. And not only that, but a man by the name of Charles Smiley did a study in the late 1800s looking at the sawdust produced by these mills. And he was showing that the sawdust produced by these mills was covering up the spawning beds in the river so that fish were not able to spawn. So not only was it a land assault, but it was a sea assault, and there was no regulation. So we really didn't have anything to stop it at that time. So then we move on to land sales. So like I said earlier, this is shortly after the Revolutionary War. The United States does not have a lot of money. They were broke before the war. They're even more broke now. They're fighting with pitchforks. So there's two main reasons they started selling land, and that was because they needed money, and also because they wanted to increase their land mass. They're the United States. They wanted to become the mogul that they are today. So they were trying to increase people to come here, and so they broke it up into the picture you see on the right into 160 acre parcels in the land ordinance of 1785. And more or less, you had to get at least 80 acres, but the price per acre then was only $1.25 an acre. To compare that to now, $1 in 1841 is about $300 today, and an average cost of an acre today is about $3,000. So it's about 10 times cheaper to buy land comparatively then than it is now. And not only that, but they were trying to incentivize people to settle here. They were trying to grow their country. So they were offering homesteads to European settlers. And what that means is they could come over here and claim one of these 80 acre parcels for free. All they had to do was live there. So they're ramping up the people in the area, they're ramping up the industry, land sales is going on. So the next thing that comes in is agriculture. Along with all these people coming in from Europe came wheat farming and dairy farming. That's what they did there, so that's what they did here. Again, this is not regulated. There's no regulations. They're able to just kind of do what they please. So, and they want to be next to water too. These are farms that need a lot of water to irrigate their crops or give drink to their cattle, and also get rid of their waste. Specifically cows. Cow manure is very rich in nitrogen and phosphorus. So you have these farms that are creating a large amount of cow manure, more than what the landscape can soak in, and then that is running off in the watershed. So now you have the lumber industry that's creating this nutrient overload in the industry up front, and then now you have agriculture tacking on top of it. And then we move on to the 1870s. So fishing, gill nets. So if you see at the right, there I have a picture of a gill net out of Lake Michigan. Those are some salmon that are being caught. And in the lower picture, you can see a gill net is just basically a mesh net that sits on the bottom of the water column. In order to catch fish, they get tangled up in the net. So in 1871, there were 450 gill nets deployed in Lake Michigan. By 1879, there were 24,599 gill nets employed in Lake Michigan. And not only that, but they were 500% more effective. So they were putting more nets in the water, and they were more effective than they were because of the technological advances in the net. So they were seeing declines in whitefish and lake trout and other fish species that they were targeting for fishing, and also lake sturgeon. So lake sturgeon, again, as I mentioned, that the gill net rests on the bottom. Lake sturgeon are bottom feeding fish. So they were commonly caught in those nets. At the time, though, lake sturgeon didn't have any value. They were thought as trash fish. They were these really hard, rigid, bony fish that you just ruined your nets. So they were commonly just gaffed out of the nets and killed and left on the shore to die. I mean, they were considered trash fish. There was no value to them, which is funny. Now they are the most expensive freshwater fish in the world. So lastly, then we move on to the paper and manufacturing industry. So John Homburg opened up the John Hoburg Paper Company in 1895 in the Fox Valley, and that just started a snowball effect of other paper companies and metal manufacturing companies and other types of manufacturing companies that all came in. And again, no regulations. They want to be close to water. And the bad part about these factories is they were next to the water, releasing a lot of these unknown chemicals, these byproducts that we had no idea what they were doing, but there was no regulation. So they were just able to dump all of their byproducts into the river, which led to some problems that we're going to have now. So now Katie is going to go into a little more in depth about what some of these problems were and how we're facing them currently.
So these historical uses have caused cascading impacts on the health of Green Bay. This especially is accentuated by recent population growth. So urbanization and agriculture contribute to many of the Bay's problems today. And the figure by the Green Bay Conservation Partners here shows the extent of urban areas in gray. And the streams in red are considered impaired by nutrients or sediment. And as you can see, the Fox River there on the southern portion of Green Bay is one of those impaired streams. So basically the impairments in these streams feed Green Bay, and they're dumping all of their problems into the bay itself. And so these streams largely contribute to the 4 issues I have listed here that I'll dive into over the next couple slides. So bottom contamination is still a large problem today, even though substances that are causing these issues have been banned for decades. And this is because these chemicals tend to latch onto sediment and persist for a really long time. And so they're very resistant to chemical breakdown and will just stay on the bottom for years and years. PCBs, or polychlorinated biphenyls, and mercury are the 2 that we probably hear about the most. And these 2 chemicals come largely from industry. So PCBs were byproducts of old paper making methods, and most mercury contamination comes from burning fossil fuels. So even though these fossil fuels are emitted as a gas, they can latch on to rain droplets and stuff in the atmosphere and come back down to earth and become problems again. PFAs are found in firefighting foam and other surface treatments like nonstick cookware. PFAs stand for per or polyfluoroalkyl substances, and they're kind of an up and coming contaminant. We just are learning about their prevalence in the ecosystems and the health impacts that they have on us as people. We hear less about other substances like DDT, lead, and arsenic, but they still exist in the bottom of our water bodies, though in lower levels. Another issue in Green Bay is the addition of nutrients. So Green Bay surrounding land uses are largely developed and heavily farmed, which adds nutrients to the water through runoff of rain and snowmelt. And when fertilizer is applied to fields, it can be washed into nearby water bodies. And unfortunately, what makes crops grow also makes blue-green algae and algae grow. And it can cause algal blooms like this one in the lower left-hand corner here, which is a photo that Brandon and I took at Metro Boat Launch, which is on the Fox River before one of our sampling events. It was kind of gross. And sometimes these algal blooms can get so horrible that you can see them from satellite imagery, like here in 2013. So when all this algae dies at the end of every summer, which it does because we live in Wisconsin and there's winter, it settles down to the bottom where bacteria decomposes it. And this decomposition process uses oxygen, and lots of decomposition can deplete oxygen near the lake or river bottom, which causes a dead zone. Many of you have probably heard of the dead zone in the Gulf of Mexico, and just like that one, Green Bay has a dead zone that materializes late summer, often on the eastern shore near Dykesville, which is this red splotch you can see here on this map. However, wind currents can carry this oxygen depleted water elsewhere. And so this figure is showing the dead zone by these red spots. And as you can see, it's very mobile. So sometimes, like in July of 2011, it's very localized to Dykesville. Other times, like in September 2009, it's spreading the entire width of the bay. And in other instances, like July of 2013, it's in the southern bay. It's completely localized elsewhere. And so sediment contamination, excess nutrients, and the dead zone affects life in Green Bay too. I'm sure many of you have seen fish consumption advisories like this one on the left. Bacteria in the soil integrate these contaminants I just talked about, PCBs, mercury, among other things, into their tissues. And when other things eat them, like bugs, the contaminants from those microbes are incorporated into the consumer. And so this chain continues, and each link in the food web accumulates contamination. Fish are pretty high up in the food chain, and so they can have dangerous levels of contaminants in them. And this whole phenomenon is known as bioaccumulation. When high levels of decomposition occur, oxygen can be depleted higher up in the water column, not just the bottom anymore. And this can happen really fast, which causes fish kills like this photo here. These problems, among others within the Great Lakes, have led to the development of areas of concern, or AOCs. And the creation of AOCs was largely spurred on by the Clean Water Act in the 1987 Great Lakes Water Quality Agreement. There are 31 AOCs within the Great Lakes Basin, and that's what this map is showing. The Fox River and Green Bay AOC is this one right here. And it's one of 5 in Wisconsin, and the other 4 are listed here. So there are 14 possible beneficial use impairments, we call them BUIs, that can be assigned to an area of concern. And BUIs identify specifically how the water body is impaired and address problems like habitat alterations, invasive species, pollution and contamination, among a bunch of other things. And shown here are the 14 possible BUIs that can apply to an AOC. Displayed in red are the ones that apply to Green Bay. So as you guys can see, there's quite a few of them. There's 12. And we have a lot of work to do. But thankfully, one of them tainting a fish and wildlife flavor, it's this lighter gray one near the top. That was recently removed this year. So we're making steps in the right direction. And so a lot of the restoration projects that are being done right now in Green Bay are addressing these beneficial use impairments. So here's a couple of them. Perhaps the most notorious due to its cost and duration is the Fox River dredging. The dredging effort removed PCB contaminated sediment and capped areas where dredging was infeasible. The entire effort took 17 years and cost over $1 billion, which made it the largest dredging effort in history. And the dredging setup is shown here in the bottom right. A vacuum sucks up the contaminated sediment and it's pumped to a dump site on land. And although this method is very pricey, the effort is an imperative baseline step to improving the health of Green Bay. And I think a lot of projects that come will be examining how this dredging effort has affected life in Green Bay. So best management practices are another tactic that are heavily being advocated to reduce nutrient inputs to the watershed. They are perhaps the most important, but they're also one of the hardest tactics to implement because they rely on private land ownership. BMPs include practices like planting and maintaining native vegetation along a lake to keep soil on the land rather than it eroding into the water. Exercising no-till agriculture to retain soil on the land. And there's a ton of these things. You can look them up online. Landowners and farmers can do a lot to help keep their watershed healthy. The Cat Island restoration effort is unique to Green Bay. The Cat Islands used to be a chain of small islands in southwest Green Bay, but they were washed away by high water levels, storms, and waves in the 1970s. These islands provided a ton of fish and wildlife habitat, and they protected the shallow waters and wetlands behind them. And so their loss really magnified and contributed to Green Bay's problems.
Katie, we're not seeing your slides advance anymore.
Oh no.
It's because you haven't advanced slides in a while.
Oh my goodness gracious me. They're there.
I just think she thinks you should be advancing slides like on that one.
Okay, yeah, Age of Restoration slide, we should still be there. You guys seeing that one?
Yep.
The Age of Restoration at the top? Okay.
Yes.
Cool. Sorry, this one is a tangent. So the loss of the Cat Islands really contributed to a lot of Green Bay's problems. So in the 2010s, the islands were reconstructed and connected with a dike composed of clean dredge material from the maintenance of Green Bay Harbor. And now these islands house a multitude of shorebirds, including endangered piping plovers and whooping cranes. So definitely a success story. And the last couple points here, the University of Wisconsin Green Bay is continuing to explore the health of Green Bay with the help of their sponsors. So here in the bottom left, we have Jade Arneson. She examined the usage and extent of wild rice, which is a native plant that's really important for waterfowl and other wildlife. Several other projects have looked at movement or reproductive aspects of a couple fish species, namely lake whitefish, lake sturgeon, and northern pike. And so I have to show these sturgeon photos because they're amazing. And so it's just so cool to see that fish like this are using the Fox River. These were captured as part of Stephen Tucker's graduate research by the De Pere Dam, and he's the gentleman here on the right in the left-hand photo. The one on the left was probably the biggest in the world, but we just couldn't get it officially weighed. 7 foot, 2 inches long. And that brings me to Brandon and I's research and the questions that we hope to answer about Green Bay. Kicking it off with my research, my master's research is focusing on macroinvertebrates. So invertebrates refers to animals without a spine, like insects, and macro just means big as opposed to micro. So I'm looking at big water bugs. No large comprehensive macroinvertebrate studies have been done since 1978. And as I just explained, lots of restoration efforts have been made since then. And so I wanted to see if the habitat improvements are affecting the bugs and if those existing problems like low dissolved oxygen are impacting what bugs can exist in those areas. I'm also curious where bugs, especially aquatic worms, like to live in Green Bay, because a lot of these species are pretty new and little is known about how deep of water they live in and what bottom type they like. And so I hear all the time that macroinvertebrates are kind of a weird thing to study and why do we care about them? But like fish, macroinvertebrates can be indicators of water quality. So for example, if you are fishing for brook trout, you have to go to a really cold, clean stream. And if you catch a brook trout in that stream, then you can infer that information about the water quality. And bugs can tell similar stories, and sometimes in some cases, they're even more sensitive to their environments than fish. And so to emphasize this point, pictured below here are some macroinvertebrates that I collected from Green Bay. And I'd like to point out that they're not actually pink when they're alive. We just dye them to make them easier to see in our sample processing stage. They, to the left, are good indicators of water quality, and they're labeled in green with the smiley face. They include baby mayflies and baby dragonflies, as well as native clams. And further right are poor indicators, which include midges and aquatic worms. And I'm sure many of you are familiar with midges. They're also known as lake flies, and they're the things that swarm you and really annoy you when you're trying to troll when you're fishing. But generally speaking, these organisms on the right, midges and worms, can tolerate pollution, low oxygen levels, and high nutrient levels, whereas the good indicator groups cannot. And perhaps most obviously, bugs are links in the food web as they feed fish. So if we wanted to restore a certain fish population, it would probably be worthwhile to see that their desired food source, if it's a bug, is present in that system for them. And so to answer my first question, I dug into some historical surveys, and I chose to compare my results to the 1978 survey because it's the most recent and the most extensive study. It was led by Bruce Markert, and he found 62 different types of bugs, which included 27 worms and 14 different lake fly species. Before 1978, the Green Bay bug community was examined fairly regularly, about every decade. And these graphs off to the right show trends in different types of bugs over time. So the dark green is 1952, the middle green is 1969, and the lightest green is 1978. And as you can see down here on the bottom, midges and worms, and by the way, worms are labeled as oligochaetes here, they're on different graphs because their abundances are so much higher. And in the lower graph here, worms are split into two different regions because, as a reminder, this inner label is the area of concern, and worms were super abundant there. So they split them up between the area of concern and the rest of the bay. So you can see that worms generally decrease over time, really dramatically from 1952, which suggests that the water quality has improved. And although you see off to the right here, this little bump in midges, midges were also, if you remember back, one of those other poor water quality indicators, but they're a little bit better than worms. So we take this as a sign of improvement. And this graph up on the top here are those happy faced or kind of iffy, okay groups of bugs. And some like sow bugs here in the middle or clams to the left, they spiked dramatically in the most recent survey in 1978. So I'm really hoping that these trends in my data continue. So to answer my research questions, I sampled 269 sites around Green Bay. And they span from the De Pere Dam in the Fox River to Little Sturgeon Bay. And the white dots are sites that were also sampled in 1978. There's 97 of them. And the black dots are new sites that I added. We used a sampling device called a Ponar, which is pictured off here to the left. This thing is a beast. It weighs 50 pounds empty. And so you can only imagine once you fill it with a bunch of wet dirt, how much more it weighs. And yet somehow after 2 summers, my arms are still the same size. So I just think that's simply not fair. But it's a big claw of dirt or a big claw that grabs a hunk of dirt, and then we empty it into a sieve, which gets rid of most of the debris, but it'll retain larger debris and the bugs. So we put all the leftover material into cups and preserve them with dye, like this cup that Aaron's holding here, so we can more easily find the bugs later in the lab. This is where all of our sample processing happened. I like to call it our COVID sweatshop. When COVID really kicked into gear, we were kicked out of campus, and so we did all of our work in the boat shed, which was pretty awesome because we were outside, we had fresh air, and we could social distance. So I think that's a win situation all around. And as you can see from this photo, our sample processing is a very collaborative effort. So after we collected the sample, which James is doing here, and we preserved it into cups, we then subsampled it. So instead of looking at every single bug in the whole sample, because that would take forever, we would randomly pick a chunk of it to look at. And so here, Claire is picking out all the bugs in a subsample, so we don't have to look at every single one. And the super small bugs are put onto microscope slides, particularly midges and worms, because that squashes them and allows me to see some features that I need for identification, like midge teeth, which are shown here in the upper left, and worm hairs, which are shown below that. And finally, after all that work, the macroinvertebrates could be identified. Each sample took about 7 hours to process after collection. And in this entire survey, we identified 22,317 bugs. And that's just what happened at UWGB. So a lot more happened at Lake Superior Research Institute, which took some of our samples and processed them there. So that number is not counting what they found too. A lot of organisms in this study. And so the community information that I collect is then summarized with various metrics, like the ones shown here in this bottom table. And then they're analyzed with statistics, like this graph up here in the upper right. I'm in the analysis phase now, so I'm excited to pick apart this graph, see what it means, and learn a lot from this data. And as I mentioned before, another one of my research questions was where the bugs like to live, including what bottom types and depths they prefer. And so to determine this, we mapped depth and we mapped bottom type. So the map on the left is showing that the west shore is primarily sand, that's that darker brown color, and the east shore is mostly silt, which is that lightish cream color. The darker spots show areas of zebra mussel beds, which is kind of cool because zebra mussels weren't fully established in 1978, which I'm comparing my study to. So we can really see where they've monopolized in the bay. And then the map on the right was created by one of my advisors, Dr. Chris Houghton. It shows depth contours from a mapping effort that Brandon and I conducted. And the scale off to the left there is shown in feet. So we drove around the area of concern going 5 miles an hour, and we just kept our depth finder, just like the ones on a lot of fishing boats. And it measured depth, and we also had a side scan on so we could determine substrate, what the bottom looks like that way too. And so the takeaway from this map is that the entire area of concern generally does not exceed 12 feet, except within the shipping channel. And it's shallowest over here in the southwest, right by the Duck Creek mouth. And so, like I said, my research is still ongoing, but this is some of the things I found so far. So for sure, the star of the show is worms. My sampling effort collected 114 different kinds of bugs, and 38 of those were worms. They were also the most abundant by far. Worms existed in abundances up to 11,417 per square foot. And I have a hard time wrapping my head around how many that is until I'm thinking about like standing on a foot by foot tile. And that would be me standing on 11,000 worms, just a ton. We were hoping to find lots of mayflies because they're good water quality indicators, just like the one in this video, but we didn't. So that kind of sucked, but that's okay because conditions do appear to have improved a little bit since 1978. Even though worms were found in abundances up to 11,000, they averaged a little bit lower than they did in 1978. I also found fewer of those sad face bugs, if you will, and more of the smiley face ones. And if I look at only the 97 sites that I shared with 1978, we found 71 different types of bugs. And going back to a couple slides ago, they found 62 different types of bugs in 1978. So we found 9 more. And I'm still kind of investigating why that might be. Because in addition to that, only 41 of them were shared between 1978 and my survey. We also found that dissolved oxygen levels are low in the area of concern, just like they are in the summer dead zone farther north. These low oxygen levels do appear to affect which bugs can live in those areas. Where oxygen was commonly low, there tended to be more worms and midges. And you can see this in this map off here to the right. So the black is showing high abundances of worms and midges. And as you can see, there is a concentration right off Dykesville on the east shore where hypoxia occurs, as well as in the southwest corner of the bay over by the Duck Creek mouth where it's really shallow. So I've only begun to explore how oxygen and all these environmental characteristics affect the bugs. And I'm excited to dive into how or if bottom type and depth do as well. And as well as how my research ties into Brandon's research, which he's about to get into.
All right, so my research. So I'm doing stable isotopes, which are really flippin' cool and a little confusing, but I'm gonna do my best to describe to you what's going on. So basically, we look at two things, carbon and nitrogen. So what carbon can do is carbon can be used to track what a population is eating. So for example, you have some top predators like the muskie and the largemouth bass, who might be eating pumpkin seed and yellow perch, who might be eating oligochaetes and midges here, who might be eating one of these primary producers, which is the real question here. What we really want to know is which one of these lower carbon sources is supporting the upper trophic levels. So in these pictures here, I have sago pondweed, a very common weed in the lower Green Bay, epiphyton, which is just a green algae that grows on the surface of rocks and other plants, and then phytoplankton on the end here. So for example, if we were to have a 100% closed system in which largemouth bass ate pumpkin seed, which ate worms, which only ate sago pondweed, the sago pondweed is going to have a very unique carbon signature, and it's going to exactly match what is in eating it. So if those four things are in a system, the carbon signature of the sago pondweed is going to be the same as the carbon signature in the worm and in the pumpkin seed and in the largemouth bass. So by following that pathway, you can see where the food came from all the way from the bottom. And then we have nitrogen. So what nitrogen can do is it takes a different level where now we're breaking it up into trophic levels. Nitrogen can help us look at where an individual sits in a trophic level. So, in the picture you see to your right here, on the first example, this is an example of what we would consider a non-healthy system. So here we have lake trout feeding on primary consumers, which would be something like zooplankton, which are feeding on primary producers. So that's not good. Lake trout should not be feeding on zooplankton or small invertebrates. They should be feeding on fish. That means there's something wrong with the system. So what we would really like to see is something more like this on the right-hand side, where you have lake trout feeding on a forage fish like alewife, feeding on, here it says Mysis diporia, or some type of shrimp or small invertebrate, which are feeding on primary consumers, which are your zooplankton, which are then feeding on primary producers, which are your plants. So we can use nitrogen to look at where we lie in this spectrum, and we want it to be closer to that, that 5 trophic level area. So in order to collect all this data, first we go out and collect fish. So we use gill nets, electrofishing, and fyke nets. Gill nets, you remember I mentioned earlier in my historical slides, very effective at catching fish. Here I have a couple pictures of a log perch and a western banded killifish. And, with electrofishing, that basically we just apply electricity to the water in order to knock the fish out for us to be able to bring them into the boat, sample them, and then release them. So then we want to be able to get macroinvertebrates, so bugs. Those we use the Ponar like Katie described. And we also use rock bags, which is in the bottom left-hand corner here. You can see, which is just how it sounds. It's a bag full of rocks. We put it on the bottom of the water column, and inverts colonize it. Then we pull it out a month later, and we're able to collect those inverts and sample them. Then, of course, we got to get our plankton and plants, which are the most important part of any food web. You have to know what your basal sources are for the food web, or you're not going to be able to get accurate results. And we went out with the plankton tow net, which is pictured on the left here. And that's something you drop to the bottom of the water column, you pull it up vertically, and then everything collects into a little collection cup here, and you pour it in, bring it into the lab, and then we have it sent into UC Davis to be analyzed. All the plants are collected by hand, pretty self-explanatory. We see a plant, we grab it. If they're submerged, we use an extended rake and we kind of rake them in. And that's how we collect our plants that way. And then again, everything is all sent in for analysis for carbon and nitrogen to see the levels. So we're able to analyze them. So on the back of end of everything, we get something that kind of looks like this. So there's a lot going on here, but basically you can see the green represents plankton, the red represents periphyton, and the blue represents sago pondweed. So this is for the overall population of fishes in the bay that I sampled in the last 2 years. So as you can see, and what we would expect in Lower Green Bay, because it is such a nutrient-rich system, based on a lot of the reasons that Katie gave earlier and all the nutrient loading, that plankton is what you'd expect in this system. Now, in a healthier system, we would like to see a heavier reliance on stuff like periphyton and common plant weeds, because that usually is an indicator of a healthier system. So in moving forward, we would like to see the reliance of this balance out a little bit. You know, here we have about 75% of the diet, 75% of the diet is plankton, 20-ish% is periphyton, and 5%-ish is sago pondweed. We'd like to see that balance out a little bit more. So moving forward, so when we're all finished up with our projects, we'll be able to assess the health of the current macroinvertebrate community structure, and also see how it's changed over time and how it changes within those dead zones that Katie mentioned, those low oxygen areas. Also, we'll be able to determine the health of the Green Bay food web and compare it to similar ecosystems. So what can we use this information for, right? What are we doing all this for? We can use this information to help build a body of evidence that could potentially get the degradation of benthos BY that Katie mentioned earlier off of Green Bay. We would like to get all of the BUIs delisted off Green Bay, but specifically for our projects, we can provide evidence that'll help exactly address that issue and possibly get that removed. Furthermore, our data can be used to assess in the development of future management and restoration strategies in the Lower Green Bay. So whether that be stocking fish to balance out the food web, or adding substrate, artificial substrate for bugs to colonize, or anything like that. So what can you do? Spend some time educating yourself further and help spread awareness. These seem like there's a lot going on here, but it doesn't take much. We just need a little bit that can go a long way. You can volunteer with your local conservation organizations, such as Ducks Unlimited. You can help financially by donating to one of these organizations, or even something as small as just buying your state park pass every year. Or my favorite, if you really want to help out, stop down at your local university and offer massages to the stressed out, overwhelmed grad students working here at the university, because Lord knows we could use it. And with that, we'll take any questions.
Great, well, great. Thanks, Katie and Brandon. Yeah, we will pivot to the questions now, and we've got a few already in the chat. So I will just go into those in the order that they came in. And the first question is from Terry, and the question is, how and why does the water level of the bay rise and fall as much as 12 to 18 inches sometimes, several times a day?
So I'm not sure if you guys are familiar with seiches, but when it's windy outside, the lake kind of acts like a bowl. So the wind will push water to one side of the lake, causing it to rise kind of at a slant. And then it rocks back and forth. Like, just kind of imagine having a bowl that you're sloshing back and forth. That's exactly what the lake is doing. And so that can cause, if it's really windy, drastic rises and falls in water level.
A lot of times south winds are matched up with lower water because it's pushing the water out into Lake Michigan, and vice versa, north winds are associated with higher water. Not always, because there's a lot of other factors at play, but that's a good indicator.
Okay, thanks. And we've got another question here. This is from Alyssa. The question is, do you think that it's possible that the macroinverts could be bioaccumulating some of the contaminants like PCBs and then passing those chemicals up the food chain?
Definitely. Yeah, things that just live in the bottom of Green Bay and the sediment in general, where those contaminants lie, they are whatever's living down there. I think I use the example of bacteria, but what bugs will eat bacteria, and then the fish will eat the bugs. And so when fish will eat not just one bug, right? They'll eat a lot. And so all of these bugs will have little trace contaminants in them. And so when they eat like 10 bugs, they're incorporating the contaminants of those 10 bugs into themselves. So that's the whole concept of bioaccumulation.
Sure. Okay. Next question that we have comes from Brad, and the question is, it says, so the last survey of macroinvertebrates was done around 40 years ago, right? When would you recommend that the next survey be done? In 2060? 60? Question mark.
Next year.
I love that comment. I don't know why there was such a big gap. And let me clarify that there has been smaller bug studies done since 1976, but the one in 1976 was by far the largest and most comprehensive survey, like looking at the entire bay as a whole or a large chunk of it, just not one specific little section. So I'd like to go back and see the trend of these comprehensive surveys go about every decade, but I guess that will depend on funding and people's interest.
That's really a big problem in sciences nowadays, is that there's commonly a lot of funding up front to start the project and not a lot of funding to continually monitor it after the original project is concluded.
I do think with that dredging effort and everything, people are going to be interested how that dredging has disrupted that whole dredging effort is like removing substrate and sediment from the bottom of the Fox River. So it's disrupting everything that's living in that too. And so I think there are going to be a lot of studies that we'll be looking at and a lot of interest in looking at how macroinvertebrates, among other smaller organisms, are going to recolonize this new habitat.
Okay. The next question comes from Karen, and the question is, were rock bags part of Katie's sampling or just the Ponar grabs?
Just the Ponar grabs. Rock Bay, Katie was focusing more on population. So you can't really, with a rock bag, you can't get an accurate population estimate from that. With a Ponar, you can, because it's a known amount of soil that you're taking, and you can calculate that into a metric. With rock bags, you can't do that. But with my study, I'm not concerned about populations. I'm just concerned about, where they lie in the food web. So I didn't need those population metrics.
And the two different methods target different bug communities too. So what I'm collecting are things that will burrow into the dirt and live there. But different kinds of insects will colonize these hard substrates like rock and wood, or even like the edges of docks and stuff. Those will be totally different bugs than what lives in the dirt.
Okay. Okay, and then the next question comes from John, and I may pronounce this incorrectly. Have you found any Hexagenia mayflies?
So we found one and half of one, and it was at the mouth of the Suamico River last year. We were hoping to see a lot more. The Hexagenia mayflies were the ones I showed in that little video that was on loop while I was talking about my stuff. So there was a stocking effort out there conducted by Jerry Castor, and so I was excited to see if they would recolonize or not, and I haven't. Unfortunately, I don't think they have yet.
That was another one of those funding efforts where he had gotten funding to originally do a lot of these stocking efforts for Hexagenia, and there wasn't so much monitoring money later. So there really wasn't a good measurement on success from that, other than we only found one, so probably not very successful.
Okay. The next question comes from Bruce, who mentions that he is a Dykesville resident, and the question is, tell me whether you are optimistic about the dead zone situation improving.
Oh, that is a good question. No, in short of it, because it's a two-pronged problem, which a lot of people don't realize is you're getting dead zones from agricultural runoff from the river that's causing a lot of phytoplankton bloom and sucking all the oxygen out of the water. So that's one. So we can attack that issue. That's something that we can understand, and we can help with. But the other end is cold water intrusion from Lake Michigan. So basically, hot water will sit on top of cold water. So as the cold water from Lake Michigan moves in, it's not allowed to mix up. So everything that's on the bottom sucks all the oxygen out of that water, and that creates a larger dead zone further out in the bay. So that I don't know that anyone really has a handle on how to approach that problem, and that may be something that's been here longer than we realized. But I think that there can be some things that can be done for stopping dead zones from the bay, but it really has to start with getting a lot of people on board with stopping non-point source pollution, such as runoff from farm fields and fertilizers and stuff like that, because that's really, we can't make you stop doing it at this point. So if people don't help and proactively take the initiative to stop putting fertilizers on their lawns, and even on a small scale, that is very helpful then. So I think it'll take some big change on a law standpoint before we'll see movement in that direction.
Okay, let's see. The next question comes from Karen. Were there species that you didn't find in the population sampling then that the rock bags collected versus the PONAR?
Oh yeah.
Yep.
So yeah, as Katie mentioned, they're selecting for different things. So just off the top of my head, amphipods, which are freshwater shrimp, those, they don't burrow, so those are more common in the rock bags. Isopods are more,
which are like those pill bugs you find
Yeah.
on land.
Yep, they're more common in the rock bags. Zebra mussels, we also get attached to the rock bag substrates. Whereas in Katie's samples with the ponars, you get stuff like the larvae of midges and the larvae of other bugs, and chironomids, and a lot of chironomids. I get chironomids too in the rock bags, but they're much more abundant in the ponars because that's where they live.
Okay, let's see. Here's another question or comment. It comes from Jade, and it says, "Great job. Brandon, could you explain why you chose sago pondweed specifically? I'm guessing because it's quite predominant."
I'd actually like to know this answer too.
Yeah. So to be perfectly honest, that I'm still working on whether that's going to be the final model. That's just my guess based on visually the bay. I spent a lot of time on the bay, and there's two things that really dominate the bay for submerged aquatic plants, and that's sago pondweed and Eurasian water milfoil. So Eurasian water milfoil is another one that could be. Honestly, I hope it's not, because it's an invasive, and I really don't want to run a food web study on an invasive. But it'll really be, as I start to work through this modeling stuff, it'll just be kind of what makes the most sense. But more than likely, sago pondweed. It's very prevalent. It's everywhere in the bay. So that's why I chose that for that specific model.
Okay. We have another question here, which comes from Claire, and the question is, does the bay historically have dead zones since it's so naturally shallow, or are the dead zones a phenomenon of agriculture?
That's a great question. I think some years in the past it could have occurred naturally. However, all these recent urbanization and agricultural land uses that have dominated this ever-growing populated watershed have only accentuated that. So maybe it would have happened pretty sparingly, definitely not every year like it does now. But the nutrients that are continuing to be added from how much agriculture is going on in this watershed that wasn't there historically is definitely making this a more regular thing.
Okay. Let's see, I think we are caught up with the questions at this point. So other people out there, if you have a question, feel free to jump in. We'll just give people a minute to collect their
If you
thoughts.
asked the one about MIBI.
Let's see, I may have not seen that. Would you like to read it, Bonnie?
Sure, from Eric. This might have been covered, but are you using MIBI to quantify the macro invert diversity? Also, do you have any recommendations for frequency of monitoring to avoid large gaps in the data?
Yeah, so I guess I'll start with that other question since it was already answered. I think about 10 years would probably be reasonable for how often this should be surveyed. As for MIBI, kind of like what we were talking about with ponars and rock bags, different sampling methods will target different macroinvertebrate communities. So when IBIs are developed, you have to pick a certain sampling method and run with it. So the IBIs developed for large rivers of Wisconsin, so like what would apply for the Fox River, uses a sampling method called Hester Dendy's, which is a passive sampling method that we just take a bunch of stacks of wooden plates and leave them in the water for about 6 weeks, and then bugs will come and colonize that over the course of 6 weeks. And then we pick them up, scrape all the bugs off, identify them, and then with that method and the things that colonize those Hester Dendy samplers are what we can use an IBI for. There hasn't been one developed for ponar sampling and things that live in the dirt yet, to the extent of my knowledge. So that's definitely something that could be done. It's a little bit over my head where I'm at right now in grad school. I would love to help out with that someday. But so hopefully that's a direction this can take in the future. And I guess I should mention, for those of you that don't know what an MIBI is, it's just, IBI stands for an Index of Biological Integrity, and it's just a score that can be used to assess water quality of a water body. So instead of picking apart all the pieces, it'll just spit you out a score, and that score will tell you if the water quality is good or bad.
Okay. We have another question here, which comes from Vicky. What are your findings about fingernail clams in the lower bay, which used to be dominant in the area?
Yeah, so I haven't dug into that as much yet. I've been focusing mostly on midges and worms because those are the dominant species in the bay. So I haven't gotten to fingernail clams yet. But from what I'm noticing, I think the abundances will be similar to that of 1978. And the same diversity. We have 3 species of fingernail clams that live in Green Bay.
Let's see, we have just a couple minutes left. So if anybody else would like to jump in the chat, feel free. We'll give it just a minute here. People might be collecting their thoughts. Okay, I'm not seeing anything else. I guess then I'll just put in a word for our next Lake Talk in this series. We've got one coming up on November 12th, which is called How to Eat Wisconsin Fish. It should be a fun one, and it features Dr. Titus Seilheimer, who is Wisconsin Sea Grant's Fisheries Outreach Specialist, and he's based in Manitowoc. I'm gonna just take another quick look at the chat, see if there's anything new in there with questions. I don't think so. So I just want to say thank you to Katie and Brandon for doing this. I really appreciate their time. We had actually planned to do this physically in Green Bay in the spring, and unfortunately had to cancel, because of the pandemic. We planned this long ago, and obviously things happened. And so I'm so glad you could join us online tonight. Thanks so much for sharing your research and taking these questions. It's much appreciated. And then just a reminder to everybody who's watching, we will eventually put this on the YouTube page for Sea Grant. It will take a little time because we will caption this before we put it online. But again, thank you so much to our speakers and to everybody who joined us tonight. Oh, and I think I do see one more question. Oh, it's just a thank you. Thank you for this informative update, and I agree. So thank you so much.
Yeah, thanks for having us.
All right. Good night, everyone. Bye-bye.
Bye.